morning, everyone. Really pleased to, I can't see all of you, but see some of you arriving in the Zoom room to hear the talk this morning. I'm just going to give a couple of introductions and say a few words before we get started and hand over to our speaker. Many people may already know me. I'm Professor Christine Rains. I'm at the University of Essex. And it's a particular pleasure to be here today because I'm a former president of the SEB and also former editor-in-chief of the Journal of Experimental Botany. So it's a kind of double pleasure to be here. And so that, I think that's enough about me. I'd really like to focus on our speaker this morning, Alicia Perera-Castro. Alicia started off our studies as a biologist in the University of La Laguna in Tenerife, and studying plant sciences. And then she's made a trip, not quite around the world, but to several other institutions, including Jena in Germany, where she did some molecular ecology studies during her degree, and then went on to do a PhD in the lab of Professor Juan Flexes on the island of Mallorca, and kind of focusing on ecophysiology. And I think that's a theme that might have run through Alicia's kind of biological life, I think, is the way I put it. And actually is now currently has returned back to take up an excellent research opportunity back at the University of La Laguna, where she's now doing a postdoc. I'm sure I've forgotten to tell you some other things, but maybe that's as good enough an introduction for now. And just before we go ahead and introduce the actual talk itself, just something to note is, if I know you'll be really excited to ask questions, but if you could make sure you either wait until the end of the presentation, where we'll have up to 15 minutes time to ask questions, or alternatively, you can pop it in the chat. And I'm really keen that any early career researchers we have in the Zoom room come forward. We really want to be inclusive, and we would like to hear from you first, any questions that you might have. So I'm going to press on and introduce the presentation. You can see this on your screens. What drives photosynthesis during desiccation? Mosses and other outliers, from the photosynthesis to elasticity trade-off. Alicia Perera-Castro, over to you. Thank you very much.
Thank you, Christine. Thank you all for assisting to this webinar. First of all, I would like to thank the Society of Experimental Botany for giving me the opportunity to give more visibility to the results of this study and to give deeply into the discussion of some hypotheses and ideas for future experiments that the results from these observations, so that at the end of my presentation, all those who want to make a contribution or ask any question can have plenty of time for doing it. So, sorry. So first, let's introduce some relevant concepts regarding the anatomical factors that are affecting photosynthesis and water relations. This is a figure of a hippostomatous leaf. This is the stomata. Here you can, you can see the stomata through which CO2 and water is being exchanged between the environment and the mesophyll. The aperture of these stomata and their density will determine the stomatal conductance of the leaf. So once inside the stomatal cavity, CO2 diffuse through all internal air spaces of the mesophyll. And if we zoom out, for coming into the cells, imagine these are chloroplasts, right? The molecules of CO2 must cross their cell walls until reaching to chloroplast and the carboxylation sites of rubisco. The diffusion of CO2 through liquid media is much lower than in gas and then the structure of the mesophyll aim to reduce as much as possible the length of this liquid phase. And this is the reason why both the thickness of the cell wall, okay, and the total surface of chloroplasts that are facing the internal air spaces are tightly related with the final mesophyll conductance of CO2, which together with the stomatal conductance and other intrinsic biochemical characteristics of the cells are determining the net CO2 assimilation rate, and it is the photosynthetic capacity of the leaf. The pathway followed by water is in most cases shared with those of CO2. The water reaches from the roots to the mesophyll following the xylem with more or less difficulty according to the structure of the conduits and the vein density of the leaf. Both characteristics will determine the hydraulic conductance of the xylem, the xylematic hydraulic conductance of the leaf. And the same mesophyll that supposed an impediment to gas and liquid diffusion of CO2 is also affecting the diffusion of water with a different strength, limiting the out xylematic hydraulic conductance and as a whole, the total leaf hydraulic conductance of the leaf. The release of water is something unavoidable to the entrance of CO2, and that's the capacity of storing water and mobilize it, the capacitance of the leaf, okay, is thought to be linked to the evapotranspiration and the plant can achieve and maintain. From this figure, it's evident that structure and that the structure is determining the physiology of the leaves and the photosynthetic capacity of the leaves. So in that context, one of my colleagues, Miquel Nadal, made a compilation of data and discovered that there is a trade-off between saturating CO2 assimilation rate and the elasticity of leaf pulling together angiosperms and ferns. This elasticity here is represented by bulk modulus of elasticity calculated by pressure volume curves. Please do not confuse with that parameter because low modulus of elasticity correspond to higher elasticity actually, and higher modulus mean high rigidity. I will remember this every time. Do not worry. This trade-off means that the highest photosynthetic rates are found only in the more elastic leaves. The basis of this trade-off is still under debate, but two main hypotheses have been suggested. The first hypothesis, exposed also in the ecology letters papers, consisted in establishing that one of the main structural components that determine elasticity of the leaves is the thickness of the cell walls, among others. If this is true, then the link to photosynthetic rates will be easy to understand, since the thickness of the cell wall is an impediment for CO2 diffusion, as we see before, limiting mesophyll conductance and therefore photosynthesis. Okay, the second hypothesis, explained more deeply in this review, defend a scenario where the link is not mechanistic but much more indirect. Elastic leaves correspond with leaves that present higher capacitance. It is higher capacity to store water and exchange it under water deficit, with the consequent big variation of the volume of cells. According to this hypothesis, high capacitance can mean high buffering of the amount of water under high transpiration, avoiding the interruption of the flow of water through the xylem and stomata. Therefore, this buffer mechanism is evolutionarily selected mainly in plants with high hydraulic conductance, stomatal conductance, and then photosynthetic rates. In our work, we did not aim to select definitively the correct hypothesis, but in this context, we had the idea of checking what were happening with the species from other phylum, species that present also C3 metabolism, but with other Balears structure and physiology. And then we aim to include this graph, that data measure for several species of mosses, both leafy and thalloid liverworts, one handwork, one carophysian algae with the addition of some Brachyophytes like filmy ferns, mainly Hymenophyllum species, and ferns. In summary, these are the main structural differences of bryophyte, carophysian algae, and filmy ferns compared to angiosperms. First, with cell walls much more thick, compensating the lack of cuticles and the fact that the structure of leaves is much more simple with one single layer of cell, at least of photosynthetic cells. And furthermore, this group of plants lack stomata, and CO2 and water is exchanged through the cell wall of all the surface of the tissue. Being the water transport may be mainly external and not through a specialized vascular tissue, even in filmy ferns, which also lack stomata but retain a very simple vascular tissue. Ferns Alice, on the contrary, present the common traits of a vascular plant with relatively thin cell walls of a common mesophyll and the rest of elements that allow an endohydric water transport, isolation of tissues thanks to the cuticulae, passive stomatal conductance, and developed vascular tissues, similar to the structure of a fern. What we observed was that all bryophytes, filmy ferns, the carophysian algae, and the two species, at least two species of lycophytes, Selaginella and Upertia, present at the same time high elasticity, high elastic tissue, and low photosynthetic rates. Only the lycophyte Illes Balears and two additional measured angiosperms behave like an angiosperm. Furthermore, even within bryophytes, any association between both parameters were not found. So we can understand the position of this group of plants in the x-axis. In angiosperms, the bulk tissue accumulation can determine the robustness of the leaves. I mean, the leaf mat area of the leaves. Yes, and that has been related with the elasticity of the leaf. At least for bryophytes, carophysian and filmy ferns, despite the thickness of the cell wall, the low cell walls accumulation together with a different cell wall composition is possibly enhancing the elasticity of their tissue, explaining their position in the x-axis of this graph. The point here is why such low elasticity is associated with high photosynthetic rates in angiosperms and not in bryophytes, and why neither within bryophytes. So in the context of the first hypothesis for explaining this trade-off in angiosperm and ferns, being or not cell walls and its bulk accumulation one of the main components of the elasticity of cell wall of bryophytes. I mean, cell walls of bryophytes seem not to determine mesophyll conductance and photosynthetic capacity in the same strength as for angiosperms. And this is a matter of debate since the thickness of cell wall have been thought to be one of the main constraints to CO2 diffusion, together with the limited surface exposed to air. But using the compilation of data that was done by Fletches recently, it is seen that actually the linearity of the decrease in photosynthetic rates, which with the increase of cell wall thickness of tracheophytes, is completely lost in bryophytes. Can you see it? I mean, there is not a significant positive intercept in this relationship. Oh, I'm sorry, in the slope, positive slope of this relationship. And the explanation of this could be that the chemical composition of cell wall and its assembly, for example, affecting its porosity is variable and much more relevant than the thickness in affecting CO2 diffusion. And probably the same is also happening for Cara algae and filmy ferns, for explaining the lack of the relationship between photosynthetic rates and elasticity in these groups of plants. However, more uncertainties exist about lycophytes, the red dots here. For them, this hypothesis cannot be applied since they present leaf mass area and thickness of cell wall in a similar range of values than described for ferns, but present the modulus of elasticity of a bryophyte. So let's go to the second hypothesis presented for angiosperms, the one that suggested that the evolutively association between high capacitance and high evaporative demand. What we can observe is that the relationship between the modulus of elasticity and capacitance is negative in lycophyte, as described for ferns and angiosperms, but for bryophytes, the increase of capacitance is completely independent from the elasticity. And it doesn't matter, the bryophytes present capacities extremely high, photosynthetic rates are not enhanced, and the storage of water does not seem to carry any diffusional hydraulic advantage in these poikiloidic plants. So for this group of plants, the priority maybe is to avoid desiccation and not to promote gas exchange and therefore water lost through cell wall. Is the lack of this evolutive constraint what is explaining the disconnection between elasticity and photosynthetic rates in poikilohydric groups? Okay. For lycophytes, something weird is observed. The negative relationship between the modulus of capacitance is maintained for the studied lycophytes, but only Isoetes velata. Okay. Only in Isoetes velata, this high capacitance is associated to high photosynthetic capacity, being the other two species, Elaginella and Uperstia, behaving like a bryophyte. It seems that for Isoetes, the evolutive role of capacitance allowing a determined non-risky evaporative demand seem to be accepted, but not for Elaginella and Uperstia. Leaf structure of this lycophyte and ferns seem not to differ considerably. However, in the cladistic study of Kendrick and Crane, it is highlighted the differences between Isoetes and the rest of lycophytes regarding the rootlets anatomy. The roots of Isoetes, as they explain, present secondary growth with large central cavities surrounded by cortex. On the contrary to the rest of lycophytes, which present a much more simple structure. where these differences could be relevant in explaining the disconnection between photosynthetic rates and elasticity in some lycophytes remain completely unknown. But one way to know it is to test if the hydraulic conductance of out-xylematic tissues is more linked to the total leaf hydraulic conductance than xylematic hydraulic conductance. According to this hypothesis, in Isoetes, the mesophyll and the stroma are the main factors limiting the total leaf hydraulic conductance, as have been described in angiosperms. But on the contrary, in Selaginella and Upersia, is water supply and xylem, which are mainly affecting hydraulic conductance and therefore photosynthetic rate. That will be an interesting experiment for doing someday, maybe after some advances in the technique used for measuring the out-xylematic and xylematic hydraulic conductance. Yeah. What we can conclude so far is that the components affecting elasticity of tissues and the structural limiting factors of hydraulic conductance and photosynthetic rates are highly diverse among land plants. Exactly. Despite this, what we have observed is that all of them present models of elasticity with the same functionality during desiccation. And we define the relative water content that correspond with the cessation of photosynthesis. It is the intercept of the x-axis of the relationship between assimilation rates and relative water content of detached leaf. And we use that parameter for quantifying isohydry and anisohydry in a continuous range. And we observe that a linear positive relationship exists between this isohydry index and the bulk model of elasticity, so that more elastic tissues sustain positive assimilation over a wider range of relative water content, whereas rigid leaves will quickly cease assimilation. Thus, this is a general mechanistic basis that explains the full range of leaf response to water stress, even in species lacking stomata, suggesting that at least in strong water stress, it is elasticity and not stomatal closure, which is finally leading to full cessation of photosynthesis. So, well, this is everything. What I would like you to remember from this webinar is that, well, structure and photosynthesis is tightly related, although the trade-off between photosynthetic capacity and elasticity is not generalized among all land plants, and even not among all tracheophytes, as we observe for lycophytes. For bryophytes and other poikiloidry groups, we can explain the lack of association between elasticity and photosynthetic following the two hypotheses used for angiosperms. The lack of influence of cell wall thickness and the capacitance of tissue on photosynthesis is explained in the dissertation of bryophytes in this trade-off. Although the reason of this independence is not well understood, and maybe cell walls have this different composition affecting their porosity and CO2 diffusion, and maybe capacitance is enhancing water retention and not evapotranspiration. But yeah. And for the studied lycophytes, only isoetes behave like the angiosperms and ferns studied by Nadal. For the rest of lycophytes presenting higher elasticity and capacitance do not affect to their photosynthesis, maybe for being xylematic hydraulic conductance, which is limiting evapotranspiration and not cyanomatic hydraulic conductance. And the last but not the least, very interestingly, all the modulus of elasticity measured for species of all phyla seem to present a common functionality that is associated with the isohydric character of the species, fully independent not just of stomatal closure, but even of the presence of stomata, as in bryophytes, algae, and filmiferans. So, thank you for listening. Now, if any of you have any question, any contribution, any idea, please go ahead. My colleague Mikel Nadal is also attending this webinar. If you want to make any comment or intervene at any moment, of course, go ahead also.
Okay, thank you very much indeed, Alicia. This leaves us plenty of time for discussion and questions if people should have them. I just want to encourage any early career researchers to come forward and ask questions if they would want to. But please, if you are, I guess, have been longer in the process of your career, please don't hesitate also to ask questions, but I'd like to get the early career researchers and give them an opportunity. So if you could use, I think the hands up function is working, and if you go into the reactions, you can put your hand up if you want to. If we could come off the screen and the people, we could see them. If you could come off full screen now, Alicia. If you come away from share screen, then I can see people, but please do use the chat function too. That's great. But if you use the reactions, then I can see more or less. Actually, I've got everyone on screen now, so if you want to raise your hand. I've got one question from Sonia. Sonia, I'm not sure if you are an early career researcher or not, but I think since we've not got any other questions just now, there's a question about how you actually measure. Is it the turgidity of the leaf, I think, is maybe what the question is. Alicia, do you want to take that question?
Sorry, I then.
So how do you measure the leaf turgidity? I think that is it. Turgescence.
Okay. So yes, I have an additional slide for the methodology because it's something quite complicated. I don't know if I can share again.
Yeah, please go ahead and do that. Yeah,
because I just, if the people were interested in this, I just save it for the end. So the point here is, well, we measure the bulk modulus of elasticity using pressure volume curves. I mean, there is another methodology that you can use for measuring this elasticity, and some tests, the biomechanical test, directly broken the leaves and calculating how is the distance of this deformation of the leaves divided by the strength that you need, the newtons that you need for breaking the leaves. This is actually a more direct way to calculate the elasticity of that leaf. But what we do here is to use the pressure and volume curves. That well, it's just okay. It's just well, the methodology is quite simple. It's just during the iteration of the sample, you measure alternatively the water potential of the sample, and you weight the sample so you can calculate the relative water content, and you build these pressure volume curves that allow you to detect which is the turbo loss point, and from this point you can calculate a lot of parameters, including the pressure potential, and you can the slope of this pressure potential against the relative water content is the elasticity. This is the definition of the elasticity of the bulk modulus of elasticity according to the pressure and volume curves. But as I tell you before, there are a lot of methodologies for doing this, not only this one. I don't know if that solved your questions.
Thank you, Sonia. Does that answer your question for you? Are you happy with that one?
Or maybe you.
Yeah, thank you. Okay. Any other questions? I don't see anything in the chat. Okay. From Vanshika Gupta, what are the control parameters when you conduct the pressure volume curves, Alicia? What are the control parameters when you conduct the pressure volume curves?
Well, each species is quite different. So, for example, if you, I mean, there is no, you have to adapt the times of the hydration during the pressure volume curves. For example, if you are working with the thalloid liverworts or with the hornworts, it's quite time consuming to let them to dehydrate little by little during the day, and you measure it little by little. But for normal moss, the dehydration is quite quickly, so it's just adapting the measurements to each sample. But the only thing that you measure, that you can control, is the water potential that you measure, for example, in a psychrometer, and the weight of the sample. This is the only thing that you can monitoring during the dehydration. The other conditions are not controlled.
Okay, thank you, Alicia. I mean, I'd invite people to put on their cameras if they would like. It's an invitation, it's not a demand, but you know, it might be quite nice for Alicia, particularly if you've asked a question, that she could address you back directly and see you on the screen. I should have said that before. As I say, don't feel pressurized, but I'm inviting people to put on their screens, particularly if they're going to ask a question. That'd be really kind. Thanks very much. Here's another question from Elise Fox. Are there any other traits of bryophytes that may confer resistance to desiccation and also to other potential abiotic stressors simultaneously? Thank you, Elise.
This is quite interesting question, and I tried to solve that question during my thesis, but it was quite complicated. I found a correlation actually between the bulk modulus of elasticity and desiccation tolerance. I mean, a desiccation tolerance that is measured, I mean, a continuous index of this desiccation tolerance, where you can calculate the long-term desiccation tolerance. And yes, I found this positive relationship between being elastic and being tolerant, but only for the species that were already desiccation tolerant, because for the sensitive species, sometimes you could find elastic tissues in that species, but it was not. I mean, the relationship was not positive. So the elasticity is helping the desiccation tolerance of the bryophyte for sure, but this is not the last factor that is allowing desiccation, of course, because some bryophytes have quite elastic tissues without being desiccation tolerant. So yeah. And we also found a negative relationship between the osmotic potential. And I mean, it's quite counterintuitive, because for angiosperms, what is thought is that the lower is the osmotic potential. I mean, the more negative is the osmotic potential, the better for the plant, because it's the adjustment of the osmotic. It's the osmotic adjustment to deal with drought environments. But in bryophytes, it's just the opposite. I mean, if a bryophyte needs to be more elastic tissue, the osmotic potential is more positive. So in the desiccation tolerant mosses presented at the same time high elasticity, but also more positive osmotic potential. This is what I found. And I test other parameters like the thickness of the cell wall, the photosynthetic capacity, but I didn't find any relationship with other anatomical traits related yes, with photosynthesis in bryophytes. Yeah, but this is a very interesting question. Yeah.
Thank you, Alicia, and thank you for your question, Elise. I'd like to encourage more questions from across the audience now, if anyone has anything that they would like to ask. Okay, so from Maria Elena, saying thank you very much for your talk, Alicia. How plastic are these tolerance traits against different ecosystems?
Okay, this is something that is not very known, but I tried to make an experiment. It was completely a mess experiment during the whole year in Mallorca, trying to compare the dry condition of the summer with the cold and wet condition of the winter, and of course we see a plasticity in the elasticity modulus. So of course, I can imagine that in different environments, this even in the same species, in the same group of plants, these parameters is showing very high plasticity. I mean, the modulation of these parameters, the adaptation of these parameters can be. I can imagine they can be quite high, even for the same species. But this is something that we have not. Published, because there are some measurements that are not published yet. But this is something very interesting, because if you, the idea is, if that parameter has plasticity, the key point for demonstrating that that parameter is playing an important functionality in another factor of the bryophyte, like, or another variable like desiccation tolerant. If you can measure in the same species different samples from different environment or different period of the year with different models of elasticity, maybe you can check if this is corresponding with different level of desiccation tolerance. So that kind of experiment will be the last proof or the last evidence that what the relationship that we found with the modulus of elasticity have a functionality, or is just a causality. Yes, thank you.
Great, thank you for that. And I've got a hand up from Sonia, and then I've got one more question in the chat. So Sonia, do you want to come in with your question?
Hi.
I think you're on mute, Sonia.
Oh.
There you are, that's fine. You should be okay now.
Yes. Thank you for the great presentation. And I want to ask only about, did you do any experiment, or did you study effect of salt stress on elasticity of some leaves, angiosperm, or leguminous plant like this?
No, I have not tested this, but I can imagine it will be a very interesting experiment, especially yes, for forcing the plasticity of this parameter of the plants. That will be an interesting parameter. Every environment factor, yes, salinity or temperature, or the frequency of desiccation, all these parameters that can affect to the modulation of the model of elasticity will be very interesting to do that.
Okay, yeah, thank you. Only I want to more clear for measure of elasticity is the same machines for photosynthesis. You measure elasticity, or
no? It's a different. It's a different device. I did not show the device here in the presentation, but it's a psychrometer. It's a device that is built, it's thought for measuring the water potential of soil. So you have small cuvettes where you can put a sample of moss.
Yes, and I understand. For measure, you measured for water potential.
Exactly. But yes, this is a device that is not recommended for angiosperms, of most angiosperms, and we have tested that some angiosperms, especially those one who has a very thick cuticle with like quercus or this very I know, sclerophyll leaves cannot be measured in the psychrometer, because the psychrometer at the end work with a it's dew point generation. You need to stabilize the humidity inside the chamber of the cuvette. Yes. And if you have these very sclerophyll leaves, there is no the minimum conductance of the leaf is so slow that there is no any exchange of the air of vapor of water between the sample and the environment. So the device never stabilizes, and you cannot rely on the values of that kind of samples with the psychrometer. But for poikilohydric plants and other species, angiosperms with very soft leaves like tobacco or like Arabidopsis, you can rely on this data, and you can see that the device can stabilize quite well, and yes, you can use a psychrometry only in these cases.
Okay, thank you.
Thank you very much, Sonia, for your question. Okay, so we've got another couple of questions here. One from P. Chitraleka. Sorry, you don't know, I can't see your first name, but it says, how did you determine whether stomata are open or closed?
This is quite easy when you measure with the gas exchange device, with the Lycor, with the GFS 300 in my case. You are measuring the temperature of the leaf, so you can calculate, assuming that the diffusional factors of the CO2 and the water, and with a series of formula, you can calculate the stomatal conductance of that leaf. And with the stomatal conductance, at the end, stomatal conductance is not only the aperture or how close is the stomata. It's like a combination of how open is the stomata, but also the density of a stomata. Is how easy the water is conduct, or the CO2 is being exchanging between the internal leaf, the stomatal cavity, and the external environment. But this is something that you can measure directly and very easily with the gas exchange device.
Thank you.
Well, you can. There is another additional devices that only measure stomatal conductance, but well, you can. The fundamental, the method is exactly the same. It's measuring just the differences in the water and calculated to translate it into CO2 movement. It's the same.
Thank you, Alicia. And I've got one question from Catherine Walsh, and asking about, have you looked at the influence of photosynthetic type on tolerance plasticity? Could a weak C2 cycle be engaged and having an effect? Thank you, Catherine. Over to you, Alicia, for your answer.
The influence of photosynthetic on tolerance plasticity.
Photosynthetic type, the type of, I guess you're talking about C3, C4, is it CAM, is that correct, Catherine? So the different biochemical types of photosynthesis, does that have any effect on the tolerance plasticity? And the question is, could the C2 cycle, could a weak C2 cycle be engaged and having an effect?
I have no idea. Well, actually, during my thesis, Jaume Flechas has a project where he was testing if the trade-off between being tolerant and having high photosynthetic rates exists. But I think it was only focusing in C3 plants. I don't know if another type of photosynthetic metabolism could enhance. I mean, if you are enhancing for the photosynthetic rates, I mean, I cannot imagine that there is a incompatibility between this between having this metabolism and having the factors that are allowing the desiccation tolerance. I have no idea. I have never read about that actually. But at least for C3, what the project of Journal of Experimental Botany and the measurements of some colleagues discovered was that some resurrection plants, I mean vascular plants that have the capacity to survive desiccation during well water conditions, their photosynthetic capacity was as high as any other angiosperm. So I think, I mean, this trade-off is not the trade-off between being tolerant to desiccation and have a big capacity, photosynthetic capacity. At least in C3, it shouldn't be occurring. So I understand that for C2 and for any other metabolism is not an inconvenient. I mean, it could be it doesn't have to be incompatible actually.
I mean, it might be more to do with the cell wall type or the extensibility of the cell than maybe the photosynthetic type. So maybe in CAM, might be changes there, possibly, I don't know. Okay, then, I think we're coming close to the end. I see there's one more hand up here from Jim. I don't know if you would like to come in and ask your question now, yeah. And then I think we have to close off for the session.
Yeah, great. Thank you. Alicia, I just have a question. We chat about sometimes about difficulties of measuring in this kind of mosses and lycophytes and this kind of lineages. What is your opinion? To what extent your results, well, I mean, measuring especially photosynthesis, but I suppose from what you said about the way you measure the pressure volume curves, also can make some difficulties. The small size of the leaves and other things. To what extent do you think this could affect your well, the lack of relationship or the different relation that you see in angiosperms that are more easier to measure?
Yeah, this is a key point, and this is something that we worry about before publishing the data, because most of the I mean, not in gas exchange. I mean, photosynthesis, I can rely on photosynthesis data without problem. This is not difficult to measure. Actually, it's just a little bit tricky, but you can measure it. But for the psychrometer, we were making a lot of proof just to understand or to be sure that our data of the psychrometer was comparable to those measured with the pressure chamber. And we actually, with several species, even angiosperms, we try to make a kind of correlation between the water potential measured with both devices. And that's when we realized that not for all the species. I mean, the psychrometer was not working for most of the species, actually, of angiosperms, only with those herbs with very soft leaves. But when we that's why we include, actually I didn't mention, but in the species that we analyze, we include tobacco and we include Craterostigma, I mean 2 angiosperms that we are also measured with a psychrometer, just to check, to demonstrate, okay, the psychrometer in most cases, in most angiosperms, especially the Sclerophilus species, is not reliable, the data of water potential and also the pressure and volume curves. But since for the most softy leaves of species with a very elastic tissue or very high minimum conductance, it's working. I mean, it's comparable, and the species that are measured with this psychrometer fall into the trade-off or between photosynthesis and elasticity, then we said, okay, if that species, if the psychrometer work for that angiosperms, for sure they will work for bryophytes, which are poikilohydric, and the release of water is much optimum. So I have had this concern all the time, but I think we are, for at least for bryophytes, we are right for species that have this trait, this structural yes, this structure and this capacity to release water and to stabilize the psychrometer. I think our data are correct. But I think for those who want to use the psychrometer for measuring what the pressure volume curves, at least some test must be done at the beginning in order to avoid possible biasing data. Yeah, this is a good point anyway. This is a very good point.
Thank you.
Well, thank you very much, Alicia, and thanks to everyone for the questions and the interactive session. I think it was really, really great. It's great to see that people stayed all the way to the end of the questions as well. I think it is, congratulations, Alicia. It was technically quite challenging, as we heard several times. There was questions about how you managed to do that. And I'm sure we could discuss that much, much longer. But I think I need to close the session off now and thank SEB and JXB for setting this up. And I thank Alicia for her presentation and everyone that contributed. I hope everyone has a nice day, and thank you very much indeed.